Hello, everyone. This is Jason Key at SBGrid, Harvard Medical School in Boston. So thanks for joining the webinar today. So today we've got Thorsten Wagner, who's joining us from the Department of Structural Biochemistry at the Max Planck in Dortmund. Thorsten, are you there?
Yes, I'm here.
Great. So whenever you're ready.
Welcome from my side to this small webinar. I'm going to talk about particle picking with crYOLO. And I guess we can skip the slide. This is already done.
That's good.
So particle picking and object detection and object localization. And in a common approach, the so-called sliding window approach, you typically start with examples, positive and negative examples, where the positive examples are your particle, like in this case, it's our TC toxin. And the negative examples are contamination, background, and so on. And then you train a classifier on it to distinguish both cases. And nowadays, for this kind of question, you use convolutional neural networks because they are pretty good in these for these questions. And then the common approach is to use a sliding window, which basically an image is convolved over the micrograph. It crops out small regions. It presented to the classifier, and the classifier will give a result like, okay, with this and that probability, I'm seeing a particle. And then you put this probability at the position of the sliding window, and what you get is a probability map that there's a particle. And the last step is then to detect the maxima on this confidence map. An alternative approach is the so-called you only look once approach. Instead of presenting multiple overlapping cropped out regions to the classifier, you put the complete micrograph into your network, and in the end you get a result where each grid cell is basically detecting one particle. I will go into the details later. If you compare both approaches, then for the normal sliding window implementation, you typically need examples for your particles and for your background and combination. For the YOLO approach, you only need particles. So you only have to label your particles. There's no negative example sampling necessary. For the context, the sliding window approach, you always have only the small cropped out region around the particle. In the YOLO approach, it always sees the complete micrograph. Regarding the computational efficiency, as the sliding window is overlapping, in principle, it's pretty fast with up to 6 micrographs per second. So how it is done in crYOLO? You start with labeling maybe 5 to 15 images manually, and you pass it through the network, through a convolutional neural network. In our case, it has 22 layers. And what you get is a stack of grids, where each grid cell is responsible for a specific region in the image. And each layer has a different task. So the first layer in principle asks, okay, does my region where I'm responsible for contains the center of a particle or not? If this is the case, then the second layer estimates the confidence that there is a particle. The third and the fourth layers are estimated and sites are estimating, say, cost and height. The objective function or loss function that we are trying to optimize consists of 4 parts. The first part penalizes errors in estimating the position of the particle. The second part of the error function penalizing errors of the width and the height of the particle boxes. And the last 2 layers are penalizing the confidence value for objects, so for particles, and for background and contamination. Therefore, this is splitted because then you can apply different weights and errors in picking on positions where actually it's just background. So the actual training of crYOLO is pretty easy. You put your training images into one folder. You put the box files into another folder. Then you specify these the parts of the folder with the images and with the annotations. You specify it in a config file. And the second step is to run a warm-up of your network, which is only a couple of epochs, and it basically ignores the estimation of width and the height. And then you do the full trainings. This is command number 3 here. And typically after 20 epochs, it converged to a result. And the complete training process on our machines lasts roughly 8 minutes for 5 to 15 images. If you have more training data, then it will last longer, of course. So a couple of comments to your organization of your. You can either do it like I showed on the last slide, so put your MSC files in one folder and your box files into another folder. And what crYOLO will do then, it will randomly choose 20% of your training data and use it as validation data. An alternative is to make separate folders for your validation images and annotations for these validation images. And you can also specify this in the configuration file. This has the advantage if you want to add later more images, you can basically start your training based on your first model in principle, because you decide which image as validation images. And if you use the more easier approach in principle, where you have your training folder and the train annotation folder, it will do that randomly. But if you then add more images and run it again, it will select other images as validation data, and this should not happen. A few words for the advanced parameters. You can tell crYOLO to use a specific GPU in case you have multiple GPUs. You can also train on multiple GPUs. This is supported. I have a flag called warm restarts. What it does, it basically changes the learning rate in a periodic manner. And in principle, you don't need it in using it for training the big general networks, which I will show you later. But if you train a specific model, you don't need this warm restarts flag. Another option is num_CPU, where you can define the maximum numbers of CPUs that should be used during training. You can tell crYOLO with the GPU the GPU reset is used, because per default it TensorFlow, which is the library where crYOLO is based on. So the GPU fraction basically limits the amount of memory that is used by crYOLO. Let's say if you have a very big graphic card, like a GB100 with 32 GB of memory, you don't need the complete memory for training crYOLO. But crYOLO per default will allocate the complete memory. And if you then want to run another GPU software on the same GPU, it will fail. What you can do then is using this flag and limit the GPU memory to, let's say, 50%. The last option is, and it was asked by a user, is a flag to skip the data augmentation. So during training, in principle, the network never sees the original micrograph. I'm always adding a little bit of noise. I'm flipping the images. This helps crYOLO to converge better. But some users want to try their own data augmentation and they ask for adding this option. I do not recommend to use that in a normal case, but in case you want to experiment with different data augmentations, this is the flag for you. So, and after you did the training, you get a file, this model.h5 file. And this model.h5 file basically contains all the things that crYOLO learned. So it contains the weights of the networks that crYOLO internally uses. And I guess it might be important to show you the configuration file that is used during training. There are a couple of options, but most of them you don't have to touch them. So I made these options that you might want to change. I put them here in bold letters. I will start with the first option. It's the architecture. Thesaurusnet is our latest architecture, and you never have to change that. The input size, this is the size which is used from in crYOLO during training. So your original micrographs will internally downsize to 1024 times 1024 pixels, but you don't have to care about it. 1024 is fine. It might be in cases where you have memory to make it possible to train on, let's say, low budget NVIDIA graphic cards. The anchors, if you train for a single particle, it's the same as your box size. So just your box size here. Max box per image. This is just to allocate memory internally. It's not used during prediction. Just set it to a very high number. Typically, I use 1000 or here in this example, 700. Filter is, so crYOLO does a low pass filter of your images, and in the filter option, you can specify. So I was talking about the filter option. This is the last option in this model section. And the filter option is describes the low pass filter that is used by crYOLO internally. So all your training data is low pass filter before it's used by crYOLO. And the filter option has two parameters. The one is a number from 0 to 0.5, which is a cutoff in absolute frequency. And the other parameter is a folder name or a path to a folder where crYOLO should save the filtered images. Next section is the train section, and there is this train image folder, which defines where you define the path of where your images are that you want to use during training. And you have this train annotate folder, which is a path of the annotation files. For then the next option is train times. By default, it's set to 10, and it's fine. You don't have to change it. It basically means how often the image is showed the network during one epoch. And you can if you have a lot of training data, let's say 100 images or so, you can set it to 1 because it slows down the training a little bit. But if you start with 10 or 50 micrographs, keeps the default. It's safe, and it never hurts to use 10 as the default value. It just makes the training a little bit longer. The pre-trained weights. This is the path to a model file, which is used to initialize the network before it starts training. Yes? Okay. So this is the path to a model file before it starts the training. And if it does not exist, it will just use a default model file that is shipped with crYOLO. The batch size is how many images are used during one step in training. The learning rate, this is a parameter for the optimization algorithm. Keeps the default, you don't have to change it. The number of epochs, it will not train 3000 epochs as specified here. This is the maximum number of epochs. But as I told you, crYOLO will select randomly a couple of micrographs as validation data. And as soon crYOLO recognizes that the performance on the validation data does not improve, it will stop the training. So it will never train 3000 epochs. This is just a maximum. So typically you train 20 epochs and that's it. Warm-up epochs, let it to 0, it's fine. Object scale, these object scale, no object scale, coordinate scale, and class scale, these are the weights for the errors. Keep the default, they are fine. Save weights name, this is your final model name. So during training, it will save your model using this name. Debug and log pass, keep it like it is. The last section, valid. You can specify where your validation images are in case you have them separated from your training data. Needed, but if you want, you can keep them separated to be a little bit more organized, let's say. Okay, the prediction is even easier than the training. You now have your model file, this model.h5 file in this case, and your configuration file. You specify it as parameter in the crYOLO predict command. You specify the folder where all your micrographs are, and you specify the output folder where the results are written. And what you get are 3 folders. One is the results in the star file format. One is the result as normal box format using the normal confidence threshold of 0.3. And one is the CBox files. And so during particle picking, each particle gets a confidence value assigned. And in the EM box files, the default is 0.3. But it might be that this is not the optimal confidence threshold for you. In this case, the CBox files become handy. You can load them and change the threshold on the fly, decrease it or increase it. And yeah, if you increase it, you will get less particles, of course. And if you found your optimal threshold that you like, you can write new box files and use them for further processing. A couple of words to the advanced parameters of the prediction command. There is this -t option where you can specify another confidence threshold that because you can generate it again using the CBox files. The next option is the distance filter. Only want to pick particles with a specific distance. You can specify it here in pixel, and crYOLO will remove particles which are too close to each other. But if your training data contains a specific pattern that you did not pick into, let's say, two crowded regions, or to pick in those regions, because it always sees the complete context. The next step is a prediction batch size. This basically specifies how many images are used in parallel. Default is 3, and at least on our machines, I cannot see any performance gain if you increase this number. So stick to 3, it's fine. And again, you can select which GPU you want to use for prediction. So the nice thing is that the prediction is pretty fast, 6 micrographs per second, as I already told you. And this makes it possible to apply the prediction command also without a GPU. So if you run crYOLO on a CPU to predict or to pick your data, it will roughly pick with 1 second per micrograph, at least on our machines. We have some, let's say, low-budget workshop laptops, and on these machines it takes 5 seconds per micrograph on a common CPU. Good. A couple of examples where we applied crYOLO. So with basically the same configuration. So you don't have to adapt it too much for each example. You just have to specify your box size and it works for very big viruses. It works on very crowded particles, but you can also pick particles very specifically. And here you see HIV spikes, which are attached to liposomes, or MPGs attached to liposomes. And in both cases demonstrate really well how crYOLO can learn to pick very complicated settings. Another nice property of crYOLO is that it's pretty good in ignoring contamination. So as far as your training data contains contaminations, crYOLO will learn to ignore it. And the last feature I would like to mention is the training on sparse data. While it might be ideal for training micrographs to completion, it still works very good if you don't do it. And to illustrate that, I run the following example. I took our toxin dataset, which was picked to completion, and I removed 80% of the particles. And you see it in the lower left corner, only 205 particles on 10 micrographs remaining. And then I applied and I trained crYOLO on this sparsely picked training data and applied the model again. And if you use the default threshold, you see, okay, it does miss a lot of particles. But if you then use the CBox files and reduce your threshold to 0.14, you get all your particles and it still avoids contamination. What you see here is the so-called precision recall curves, and I compared basically the models which were trained on the completely labeled dataset and the sparsely labeled dataset. And what I measure is, in principle, the area If you have an area of 1, if you get a value of 1, then this is a perfect particle of 0. And what you see here, the completely picked data set, you get 0.95 as an AOC value, which works, which is excellent. But also on the sparsely labeled training data set, it works excellent with an AOC value of 0.88. I did a couple of more experiments with TRPC4 with more contaminations. Check it out, it's on our wiki page. Another thing I would like to mention is the training of crYOLO as on the training of a general model with crYOLO. So typically, for one particle, you will do several data acquisitions, maybe with different grids, different camera, a different microscope, and so on. And the model which was trained on one of these data acquisitions will likely not perform that well on another data set. So what you can do then to get a better generalization is to merge your training data for multiple data acquisitions. And the result is a model that much better generalizes. And for other data acquisitions, then you don't have to retrain your model again. We did that for cryo-EM and for negative stain. For cryo-EM, we used 32 real data sets, 10 simulated data sets, and 10 only contaminations, no particles data sets, and trained crYOLO on them. And the result is a model that it's applicable to new data sets, and you don't have to train on them in principle. So you can apply the general model to new data sets and get a pretty decent result. And we did the same for negative stain data sets. Right now it only contains 10 real data sets, but we will increase the number. But at least in our labs, this is working quite well, the negative stain general model, and also the cryo-EM general model, of course. One thing I would like to tell you is fine-tuning the general model. What you can do is the general model is trained on a big chunk of data, and it learned pretty much general features. And you can use that and fine-tune this model for your data. And what it in principle does then, instead of training all the layers in your network, it only trains the last two layers of your network. And from theory, the fine-tuning should reduce the risk of overfitting, and it's much faster. So if you have a low-budget graphic card, this might be an option for you, because it's not only much faster, it also needs less GPU memory. So this might be interesting for you, but it's still more experimental. We don't have too much experience with this fine-tuning, but it seems to work quite good. And the only thing that you have to do is you basically, in your configuration file, you specify the latest general model as pre-trained weights. And during training, you have to add the flag -- fine-tune for your training, and you can skip the warm-up phase. You don't need to warm up the network for fine-tuning. If you have your model trained, you can run the evaluation script if you want to. And the evaluation script will provide you a couple of statistics about your data set. It will tell you the AOC value. It will tell you common statistics like the recall and the precision value. A union, or in short, IOU. You can do this evaluation with two modes. So every time you run crYOLO, it will write run file into a run files folder. And this run file contains the information which images were chosen as validation and which are used for training. Using this information, it can start calculating the statistics. In case you have your validation images and your validation annotation separated, you can also run it on this data. Good. The last feature that I would like to mention here is the filament mode. So in principle, it works very similar as training for a single particle. You need different training data. You can create the box file that you need with the E2 helix boxer. And then you start, and what you get are these red boxes here. And these red boxes are then used as support points for tracing the filaments. And therefore, I'm using a mask shown here in the lower part of the presentation. I'm using this mask and convolve it with your input micrograph. And the mask is adapted to your filament width. So during prediction, you have to specify created, and it can basically estimate the orientation of your filament. And then the cryo will use this orientation to trace your filament and connecting all these red boxes, and then replace the boxes with a specific distance. All the things that I have told you now about fine-tuning a general model, about training from scratch and using the filament mode, you can all of this is described in our wiki in good detail, so you don't have to take notes. More advanced examples where you have a lot of filaments that are aggregating, and it's pretty good in skipping these aggregates of filaments. So also for let's say more advanced data sets, the filament mode is working quite good. And in the gray box, you see the command that you have to use to run the prediction. So in principle, it's pretty much the same as for a single particle. So only two things that you have to add, or three things, is telling crYOLO that it should work in the filament mode. You do that with the flag -- filament. You have to tell crYOLO the width of your filaments in pixel and your distance in the final boxes that you would like to have. Good. To summarize, crYOLO makes picking fast. Up to 6 micrographs per second is possible. It's smart because it always sees the context, the complete micrograph of your where your particle is embedded. It's easy because you only need a couple of micrographs to train crYOLO. And often the training is needless because the general model often performs pretty good on new data. And the training is tolerant. So in case you are not able to pick all of your particles during training because it's just quite hard to see them, crYOLO will still do the job. So as I already told you, there's a very detailed tutorial how to use crYOLO. You can find it on our website. You can find more technical details in our preprint, and very soon the final paper will be published. And at this position, I would like to thank Stefan Raunser, our PI, for all the support and the freedom, the complete Spire team, and also the complete department group, which did a great job in helping me to create training data for crYOLO. And with this, I would like to thank you for your attention.
Great, thank you very much. Questions, you can send them to me by chat. I've got a few here. And let me take a look. So I've got one question here. How is the downsampling handled from non-square micrographs, K2, K3?
Yeah, it's still converted to 1K times 1K. But this doesn't matter because during prediction it will do the same. So even if the particles will be then in principle squeezed a little bit during training, as long as it sees the particles in the same way during prediction, and it does if you don't change the settings, this is not a problem.
So another one. In the paper, you mentioned various ways of augmenting the data, pixel dropout, flipping, etc. What ways are there to encode these directly in the objective?
I'm not sure if I got the question.
Yeah.
So the data augmentation, yes, is used internally to get a better convergence and less overfitting. But I don't get the question regarding regarding the objective.
Clarification point is, as in put them in math in the loss function.
Okay. Sure. So the question is if we describe the data augmentation somewhere in plain math, or does I get it correctly?
I'll wait and see if I get a reply here from GW who sent me the question. If GW has a mic, I can unmic them and they can.
Yeah, I have a mic.
Oh, okay, great. Go ahead.
Perfect. Hello.
Yeah, my question is just about instead of augmenting the data to explicitly put in the assumptions that go into the encoding, like symmetries, into how the objective sees the data, so that something that is flipped appears as the same thing, and so that you don't have to augment it. Sometimes that can be done with things with the filters, with different symmetries in the convolutional filters.
Yeah.
I haven't seen anyone do that too much though for cryo-EM data.
Yeah. There is an option with a different architecture. It's not CNN. How was it called? But in principle, yeah, it might be that you can take it into account in the loss function. But to be honest, I'm not aware how to do it efficiently. So I cannot say too much regarding this.
Okay, thanks.
Sure.
So we have one more question here from just based on the approach to picking from these micrographs. They're very noisy. Pranav Shah here is asking here at HMS, are you thinking to implement a neural network-based denoising tool in crYOLO?
Yes. Yes. Yeah, we are thinking about that. We are currently working on the denoising part, and as soon as it is available, I thought to make it an optional thing that you can basically say, okay, instead of using a low-pass filter, please use my denoising network that I trained for this data set. So then you can use it in combination. Yes, this is planned.
Great. All right. Well, with that, Thorsten, thank you very much, and thank you everyone for joining. And
thanks a lot.
Thank you.